Hello everyone, and welcome to Ant Biology webinar series, presented to you by Translational Outcome Research Group, Department of Zoology, University of Calcutta. As you are already aware of, that in this series we not only focus on biology, but how biology is interconnected with various other disciplines. To all our dear audience, you can post your questions in the comments and interact among yourselves in the comments. We will convey your question to our respected speaker at the end of the session, and the feedback link would be shared in the comment session, in the comment box at the end of the talk. So please keep a note on that. And today is a very special day. In India, 5th September, we celebrate Teachers' Day. And our heartfelt wishes to all our teachers from Ant Biology team. And today we have with us Professor Dwaipayan Bharadwaj from the Systems Genomics Laboratory, School of Biotechnology, Jawaharlal Nehru University, New Delhi, India, who wants to be introduced as a teacher and an inquisitive student. So a very warm welcome to you, sir, in our platform. We are honored to have you. And our mentor and convener, Professor N.R.A. Banerjee, has sent her best wishes and regards to you, sir. And the stage is all yours.
Thank you, Akashlina. Such a nice introduction, and I know now what a wonderful webinar series and biology you are going through. So, my heartfelt wishes to all my audience for the Teachers' Day. So today I'm going to talk about, the title says it all, the ethics and its practice in biological, biomedical sciences in contemporary India, what to do and what not. Why only biological and biomedical? Because evidences are there because of this wonderful digital era. So first thing is like that, just today in Times of India, this news came. What is by that? Dr. S.K. Sarin is a famous physician scientist. He wrote this thing, during this pandemic, lesson for scientists, student, be honest. And he showed only some of those papers which are related to COVID, because all of us these days forgetting doing right science, we want to be in the limelight. So Dr. Sarin wrote that how those prestigious medical journals, Lancet, New England Journal of Medicines, they are immediately, because it's a hot topics, COVID-19, so they are immediately accepting those peer reviewed, obviously, but it is a laxity in the peer reviewed. So lots of papers coming and then retracting. So just today it came, so I thought it's apt. So I showed you this title. Very recently, if you remember, just recently means within 1 month, all over India, you are finding in the newspaper. After recall of paper of over data manipulation, NCBS storm over charges of harassment. Paper recall sparks churn in NCBS. When a paper is published but author is found guilty of misconduct, one author, just pay attention, one author, because those are the consequences I will tell you, because my entire talk towards my younger generation, younger researchers, because you are naive. We were also naive at that time. So I know basically all of these young researchers are very honest, very ethical. They want to know what the line to cross and not. And most of the time, senior authors push them to cross the line. So, it comes from the updated. Initially, the Bangalore scientists discover new iron sensing mechanism in bacteria. And then the student is discredited in NCBS study, alleges harassment, everything. And this is my opening slide, and I will show this in the last slides also. First, there is a disclaimer. All the data shown here actually found and acquired from publicly available websites. Nothing is mine. I am not cooking this data. All the judgments are you. All the pictures shown here for your evolution and your judgment. So you will evaluate and judge that what is right and what is wrong. So nothing is cooked in my laboratory. Experimental data, I hope I will come and show you in another seminar, which I promised to the main organizer of this seminar. So this data I'm going to show you, everything in the website you will find. Okay, so that's my disclaimer. This digital era, that when we were doing research at the time, there was no new generation sequencing. These we were used to do sequencing, making our own gels. And there are because I did my PhD from IICB Calcutta, so there were department dedicated to photography, drawing, everything's like that. Artist Babu, we used to go to Artist Babu, just taking the picture of the gel and then labeling of those gel with those stencils and everything. They are very cumbersome. But now we are in the digital era. So we can do wonder. We can do magic with those gels and everything. We can keep them very easily. That is wonderful. You can change contrast because contrast is not coming. You can change brightness of your gel pictures to get the real meaning out of that. You can store it in a wonderfully that you will have not to see that, oh, my negative kharab ho gaya. We need not to go to library frequently. Then information highway is there, so all the time we can, any paper we search, we can go. If paper is not available, we can send an email to our friends or ex-students. They will send you the PDF version. So information searching is just like lightning first. You can compare quickly, so that these are the things, are the advantages for the reviewers also, and that's the website I'm going to talk about. You can know the source of the research material, so all of those things. So in this digital era, we can do wonder if we desire so in a positive way. But also, like the dark side, if we want to take in a negative way this advantage, we can do that. So I will tell you that, my younger researchers, what is the limit that I will also show you these things. I will tell what are the problem, and I will also tell you the remedies. Problems plenty. So this is the website. It's called Papier. It's a volunteer website, and obviously coming out of United States, is launched in 2012 because some person thought that lots of garbage in this Papier and lots of false information in the papers. So he made this papier.com. What they're doing, there are volunteers all over the world. In India also volunteers. So they are every day, their main volunteers' job is randomly going through some papers and evaluate them digitally. And obviously digitally means that's why biomedical, biological sciences are first focus. But now physics, chemistry, math, every papers are coming, but those are the other area. I will only show the biomedical and biological research. So now, because of the digital era, that everything is possible. And also, when you are talking about the histological pictures and everything, there are certain people, they have the knack to look for those wrong thing, and they find it easily. Especially, you know that if I tell a little science, the colorblind people who can be wonderful for these comparisons, the histological slides, because though they are colorblind, but their depth capacity is very high. So that's why they can, earlier in World Wars, they used to be navigator next to the bomber pilot to show where the camouflage and other things. So this is the Papier. So you can go papier.com and you will get everything. So this is the thing. And what are the things? Just today, that if I see some generalized data, what are the Papier thing? This is volunteer based. One of the major volunteers is Elizabeth Bick. He took time off from the own lab to do this job. And just yesterday, there were 112,651 papers were there. This is the 4th September 2021. These are all up-to-date documentation I will show you. And there are logarithmic scale more papers that are older. And there are 90% papers are from biological sciences and biomedical sciences, because this digital thing, they are picking up those figures and those pictures. So paper using some kind of images. And recently, lots of volunteers from other fields of the science and technology, as I said, the physics, chemistry, astronomy, you tell it. And within this more than 100,000 papers, most papers are from USA, because they are funding, their numbers of people, they are huge. So they are. But when compared these potential fabrication papers of the total number of papers, that means density from the same country, then its number changes drastically. Then top 3 candidates are China, India, and Iran. So that means we are most corrupt. And pertinent to mention here, the Retraction Watch, they open country-specific website for these countries, because Papier is the first time. And when ultimately, I will tell you that what is going on, and then when ultimately it goes to the particular journal, and they started retracting this. So there is a retraction watch, another website, and there it's so many retracted paper from India. So they opened up a retraction watch India specific. So, but now another thing is very important. Rarely the top researchers, the Nobel Prize winners, they don't do this kind of fraud in USA or other Western countries. Exceptions are here. Very recently, I'm finding one very current Nobel laureates papers are coming one by one. But there also, I will tell you in a minute what's going on, because in India also, this more than 50% of their in the United States, more than 50% of their those false papers are researchers from again Indian origin, Chinese origin, Philippines origin, Bangladesh origins. So that Nobel Prize winner that whose papers are coming right now, and I see astonished, not surprised anymore, like those most of those first authors or middle authors are from Asian continent, Indian, Sri Lankan, and so on. And to tell you that Japan is least affected, that means you see the honesty means something. And India, most of the researchers were not even bothered up to 2017 to 18 to respond. And now that everybody is jumping, responding, because they know otherwise it will come to many places. And I am fighting this war on my Facebook page. I already posted more than 130 postings. I only picked up those Indian researchers because I want to show the students which lab to go and which lab not to go. There are ethics. So those are publicly available in my Facebook pages. You can go and check. Those are connected to the PubPeer. Now, if I see this country of origin of publication that retracted for fraud of suspected fraud, plagiarism and duplicate, then you will see this is that thing. All the time, USA coming big because their funding and numbers are huge. But you see India is substantial. But the total impact on other things, we never goes in this pie chart in this much. So plagiarism, you see that then again India is huge. And duplication of publication, you see, we are almost that if you see then UK or USA, we are more than half of those United States. And if I see those every 10,000 papers retraction rate, then you will find that India is fourth, Iran is top, Romania, Singapore, India, like these things. So you understand that what's going on. And another things to tell you, in India, most of those 90% of our top people at least having one PubPeer paper because they are doing. Now I'm telling you one conclusion before going to anything. When my paper, if I see I'm a teacher or I'm a scientist, my fundamental job is to do this. I will entirely I'll show the process. So if I find that there is an image duplication in my paper or something PubPeer is reporting, so maybe I do not know that. I am telling I'm not knowing that. But in that scenario, before blaming to my first author or someone, this onus is on me, because this is my fundamental duty to see the check every day those notebook, to sit with the student during the publication time, writing everything, seeing, comparing with those figures and everything. So I am not doing my fundamental duty. So either I am fundamentally corrupt, I am not doing my fundamental duty, so I am naive, then I cannot aspire for the higher position, or I am dishonest. There is no other in between. Only two things. If your paper are coming into the PubPeer, then either you are not doing your fundamental duty, then don't aspire for the higher position, or you are a fraud. So there is no in between. I'll show you that also in a minute. This is our process of publication. What we are doing when a paper coming, ask any PI or the PhD students you discuss with your student, you saw the raw data every time. Then you write when you see that there is something question answer is we can capable of, then first draft written by the student, then correction by the mentor, then we start writing second draft depending upon the laboratory, depending upon the mentor and student writing power, then correction by the mentor, then we do third draft, fourth draft, and then we decided we will send this paper to this particular journal, then the modification according to this requirement of the specific journal, and then we submit that manuscript to the journal. Then if they accept, then the peer review will take at least 2, 3, 4 months, And then after they will say, because rarest of rare cases without that review, it will be accepted. So there will be some comments, and then you will complete the comments, revision of the manuscript, perform in silico analysis, in vitro experiment, whatever according to the reviewer's query. Then you write the rebuttal letter, then you send the revised manuscript, and sometimes second revision, and then acceptance of the manuscript when reviewers are satisfied. That will also take another 1 month. Then sometimes they will send you the galley proof. Then you will see that final correction, if any, all the images and everything. Then ultimately manuscript published. So you see how many steps? 1, 2, 3, 4, 5, 6, 7, 8, 9, more than 20 steps. How can you miss those 20 steps if you are not callous and indifferent? If I think you are not a fraud, how can you miss? You cannot miss. These are the steps. So that's what I'm saying. There are only two options. Either you are callous and indifference, that means you are a lab manager, you are not the lab PI or a scientist yourself, or you are a fraud, you are pressurizing the student. Pressurizing students means a different thing, that you never say the students, go and do this fake data. You say, oh, your data. No, this data cannot be. This is my hypothesis, this data should be like this. So you pressurize the student 20 times, a very sharp student will be frustrated and come to you with the same data what you want to see, and that exactly happened to these NCBS cases. So problems are different. See, if this is the jail, how we can in digital area we can vertically stretch that. You see that normal reviewer cannot see this until and unless digitally you see what is going on. This is horizontal stress, this is intensity low, so see it looks like different, or intensity high, or split, or you flip mirror image 180 degree, you see totally different. So everything is possible. So you see here another problem is like that. This is that one histological slide of a plant, you see, and this thing is being used in this paper. So this is the Journal of Biology, this is a microbiology, this is another Journal of Biological Chemistry. You see that how they are showing this one and this one is the same, and this one, and see that all of those say, but it was not possible before digital era to catch, but this day we can catch. So we now know. So this guy, this lab is using the same picture in different journals, and now because of this PubPeer website and some volunteers are putting their golden time, you are finding that what is going on. End of the day is not garbage. Not only that, when you are publishing a paper in a moderately good journal, you are spending the publishing fee 50,000 to 1 lakh rupees, and then you are retracting that. And when you are experimental science, you are doing minimum 20 to 30 lakh. You spend all of those experimentation and manpower, and then end of the day it is retracted, and most of the time by the chief editor of those things. So what a loss for a poor country. So when checked, we found that already told that top American scientists are not involved, and more than 80% of this paper in America also, they are also from the scientists of Indian, Chinese and Iranian origin. I'll show you this thing. One of the major institutions that suffered heavily is Cleveland Clinic, and popularly known as the Calcutta Clinic for obvious reason, because most of those persons, they are from the Calcutta region going to the Cleveland Clinic. It's very famous for cardiovascular research. I'll show you one example. You say that one recently that we are finding in the retraction news, this laboratory, that this paper is influence of P53 in the transition of myotrophin-induced cardiac hypertrophy to this heart failure. Retracted, because say that expression of concern immediately in this cardiovascular research journal, and then following an institutional investigation about the data presenting, you see that how it is. This you find this is the splicing. That means somehow from another side, this particular band is being put here. You'll find is a clear shots of splicings are there. This is one of the nomina. And ultimately, committee ultimately agreed with the institution recommendation. So the editors have agreed to retract this article. And then you see that from the same laboratory, these are the namunas, these are the examples I'm showing how they perform day after day, day after day, day after day. You see, this is the figures. You see that same figure, they're cut pasting lots of times in different figures. So again, this paper retracted. They said that this is the same thing. Authors disagree with the findings of the Cleveland Clinic board. So it goes to the Cleveland Clinic's board. They decided that these are wrong. And ultimately, the journal's editors, they are retracting this paper. See, the total 19 papers from this same laboratory, total 19 papers are in the PubPeer. And you see that how many papers retracted? 1, 2, 3, 4, 5, 6, all the papers retracted from this laboratory. Now I'm coming to this. This is that example of Indian scientists when they're working in the foreign laboratories. Now I'm coming to our country. You see this. This is wonderful. This is the statistics. You see the statistics. You see this exactly these two papers, these two figures are same. If you see this dot and everything same, how can, this is my point to the reviewers. The reviewers must be trained. How the reviewer can miss this? This is you cannot miss that. This is from the I called it Mount Olympus, Indian Institute of Science. And if you go and see those, all the garbages are there. This is that simple question to the reviewers. How can you miss this, you moron? And you see the ghostly statistics. You see that how they are doing statistics value. That one of the PubPeer volunteer also tell that this, can the authors provide the original raw data to verify the same for all the figures in the articles? Answer is like that you find it's a horrible. Now I will coming another, and this from the same laboratories, if you see then, and then when a first paper comes to the random volunteer in the PubPeer, then if they get some malpractices, then they started all of those papers from this laboratory. And you know this, see this figure 3 and figure 5, identical images. If you see here, it's clear that this portion is cut paste with this side and this side. So all of this, you see here is this cut paste side. So this one after another paper from this Mount Olympus or Indian Institute of Science, you see this, how this thing happening. And then obviously all of those authors, not only them, they will say it's an inadvertent mistake. How inadvertent mistake can be done when I show the procedure, and this will not alter the conclusion. My first point is like that. If these pictures are not altering the conclusions, then why on earth you put those pictures in the first place? And why on earth the reviewers are reviewing those things? They could review without the pictures also, because end of the day that will never change the conclusions. You see, this is from Indian this Institute of Technology. It's a very good journal, society journal. You see this same thing. You see this flip, 180-degree flip, rotation, 180-degree flip. Hope we have all satisfied all the queries. Initially, when this paper came into the PubPeer, then they said, hope we have satisfied, these authors are saying, all the queries. Let us know if you have any questions. All the above observations will not impact the overall conclusion of the paper. It's written in the website. You can go and check. This is that URL for that. And then 2020, ultimately, this molecular and cellular biology, they retracted this paper. So they satisfied with all the queries, and it is not impacting the overall conclusion, but end of the day, patient is dead because that paper is retracted. Oh, another thing, as we move on the era of the open science and the routine disposition of the original data, there will be no hiding place. Science, do it properly or don't do it all. So it is being written in that website. So they said that volunteer ultimately said, oh, another thing, as we move into the era of open science and the routine depositions of the original data, there will be no hiding place. Science, do it properly or don't do it at all. And so this volunteer is a foreign volunteer. So what that foreign volunteer is thinking about us when our young researchers will go to do PhD with them or to the postdoc with them. So that is the huge impact you see. So now I'll come to a laboratory, a national laboratory. And that laboratory, when I research, actually somebody, at the time I was known to most of the laboratories. So I was getting many unknown mail. So in one mail, in one morning, I get an Excel sheet. And I saw, and I then immediately checked all of those papers. Oh my God, this entire institute, this is the national laboratory. You see this lab number 1, 2, 3, 4, I think. Every laboratory are having doing these things. And you see the previous director has 16 papers in the PubPeer and 2 already retracted. Current director, no more current director. He just missed the tenure because they are not telling. But when it is come to the authorities' notice that 15 papers of these current directors in the papier, and with those all of those documented proof, they didn't give that director the second tenure. So this is that one laboratory. Not only that, this laboratory, this number 1 lab will cite number 19 and 21 paper, number 31 will cite 19 and 21 paper, number 11 lab will cite these 1 to 7 labs paper. So in that way, they are check the citation index also. So this is provable, and this we can prove that that is going on. So then that retraction notice of those previous director, these are those things I showed, those 2 paper retracted. This article has been retracted at a request of the editor. So it is not that you are withdrawing, because when this thing going on, Papier, what they are doing, Papier is sending immediately to the PI that this paper has these flaws, so please respond. So you can respond, you cannot, because who are Papier? Is a third party. But Papier will wait up to 6 months, and then with all documentation, Papier write to the journal that from where this paper is there, and then journal will investigate, and journal will contact PI, and then you are bound to respond, and depending upon that, they will decide whether it is a erratum, or it will be rejected, or it will be retracted. So you see, after publication of this manuscript, the editors of these journals were made aware of the fact that the articles contain identical images that indicate different results, also with respect to a prior article. After discussion with the corresponding author of the 3 articles, led to the decision that this article must be retracted. So all of it, 3 papers are retracted. You see, so this is that our best prize in the country, that every time you see that they decide in the month of September, the Bhatnagar Award. The Bhatnagar Award is in the biological sciences. So in India, godlike stature, you will get 15,000 rupees per month, and you will get anywhere 1 lakh rupees every year wherever you will go visit, just to expand the horizon of science, you know, and you will get 50,000 rupees every year just to purchase books. So you will just lift yourself. So you see, when I research, because this Bhatnagar Award in biological science started 1960 to 2019, up to this 2020, I checked that some of the total pub-peered articles authored, that is that 101 scientists got the coveted award during this time, and among 101, 32 scientists are having at least 1 pub-peered paper in their name, and 17 scientists are having blatant image manipulation multiple papers. And some of the total pub-peered articles, they are institution wise. You can see what are who are the Mount Olympus is the maximum, and you will see those 17 artists, scientists, I would say artists, they are also many of them from those IISc or NCCS or NCBS. So this is that our best prize in biological science. So now I'll come a scientist. I'll give you an example of a top biological Indian institute. This is that one of the top biological institutes. This guy did PhD, then postdoc in a very big lab, and then now a national laboratory scientist got Bhatnagar Award. You see that entire time during PhD as a first author, manipulation paper 3, during postdoc, 1 major manipulation in the cell paper, and this particular laboratory, Sam Lee laboratory, is now in the focus and in the retraction watch. So you know what kind of garbage they were doing. And this now as a corresponding author, there are already 3 papers are there, blatant papers. So entire life is doing like that. You will see this, you see this same thing. This is stretched, this is suppressed, this is horizontal mirror image. You see the same thing, mirror image. If you see this 1 or 3, it just horizontal mirror image, just they flip that thing. So the same thing, horizontal mirror image. So my point is like that, how can it be an inadvertent mistake when you are stretching, when you are flipping, when you are changing contrast? If it is a inadvertent mistake, then the same thing will be there, na? So how come the same picture that is flipped, horizontal mirror image or stretched or those things? So these are not that simple. So you see the same person is doing, this is those when you do flow cytometer. So now manually changing the quadrants of the flow cytometer, you see that this one and this one same, this one and this one same, only intensity changes. And these numbers, whenever you tick this grid, then automatically this number will be changed if you are enough expertise to do in the flow cytometer. So these are the situations you will find the own laboratories now going on. So now I will show you the two distinct different ethical problems. So I will say this is another case. You will see that what I was worried about this writing earlier about this journal, how editors are selected in the PLOS One. This editor who handled the manuscript is from CSR CDRI, just a few kilometers from Authors Institute, because Authors Institute is IITR. And you see this, when I saw this paper at that time, it was not retracted. You will see that it is a multiple image duplication. You see this is image duplication, this one image duplication. So only 2 or 3 pictures I put it here. If I remember correctly, which I am, here at least 7 image duplication was there. And at the time I was thinking that how come this paper is being, and this that particular editor who was handling this paper, actually the PhD student of this Dr. Debapratim Kaur Choudhury. So how this is that how this nexus is being continuing on. And ultimately in 2019, after publication of this article, concerns were raised about the similarities between of the following figure panels. And the PLOS One editors retract this issue in light of the above issues, which call into the question of validity and reliability of the results. And then 2 authors disagree with the retraction, and other 3 others didn't respond. So paper was retracted. Now I will tell about those 2 Indian superstars. So one person has 43 articles in the PubPeer and already started retracting, 2 already retracted, and many more will be retracted. You see these are those photographs. These are electron microscopy photograph. You see that how many kind of image duplication is there, and this is only possible to catch because of this modern digital era. And she is the elected fellow of the all 3 national academies. She was actually as the acting director of a national laboratory. Numerous committees she is sitting just to tell the fate of you, to decide the fate of the young researchers. And actually she sits all committees right now. And this there are, I told you already, 2 retraction. These PLOS One editors retract this article due to this unresolved concern. And the PI didn't agree with the retraction. Other authors did not respond. But you see that these now papers are coming retraction, and all of those blatant thing, and you see already 43 articles because of these image duplications in the PubPeer website. Now I will tell you. So these are the seeing all of those fast authors, So sometimes you will see there are same fast authors, many. Where are those fast authors are going? If you see, this is the same laboratory. This is the fast author here. This is the fast author here. This is the fast author here. You will see this flow cytometric everywhere, those data image duplications. And you will see that where this fast author, I started to do research. Then I find in the Bethune College, this fast author is a faculty. And not only that, these all papers in her name. Still, she is not withdrawing from there. So Indian superstar number 2. So this particular person has a 48 articles in PubPeer and already started retraction, and 8 articles already retracted. You see that this is a chief scientist in the National Laboratory, membership of all editorial boards, and all of those fellowships of those academy and National Academy, Medical Sciences and everything. And you see this article has been retracted at the request of the editor-in-chief. All of those articles and multiple things, you are finding the same thing. They are putting everywhere image duplication. Now we'll come to those other things. Then I do the investigation department-wise. So this is one of the very famous department in a very famous institute, MRDG IICC. And department has 14 faculties. At the time, 11 out of 14 faculties, 78.5% are PubPeer for various reasons, with a total of 38 papers. And some of these are for careless mistakes. And these people are sitting all DBT committee, DST committee, and deciding your fates. And among these 2 papers are already being retracted. This is a little 2019 data. I'm sure if I go back and I will see that more papers are already retracted. Departmental chair at that time has 7 PubPeer paper. According to PubPeer comments, they are volunteers. These are image duplication after changing contrast, intensity, cropping, image duplication with different background, undeclared spicing, identical images with different exposure, image duplication after horizontal stretching, recycling between papers, all sorts of things, not simple image duplication. And they decide that who will be the teaching morality, ethics, and integrity, because they are in everywhere in all of those committees. Here are all of those link of those each paper in the PubPeer. Don't believe me, go see and then judge. This is that another case study. This is the unnecessary delay in response event. So this is things happens when long time ago. This is that if you see, this is the image duplication. See this image duplication. See this image duplication. Multiple image duplications in once. So when, dear Papier, we have gone through your posted comments and thanks for the query. Authors is writing 2 years back, and we are carefully looking into it. It has happened for the first time. It seems some errors, mistakes had happened during the assembly of the figure panels. You see that single 1, 2, 3, 4, 5 already there. So we'll be get back to soon. So then after 1 year of author's response, volunteer asked, we will soon get back to you on this matter. Is there any progress with that? And then just 9 days ago, the paper was retracted. So following the publication of the article, concerns were raised regarding the results presented, and ultimately the PLOS ONE editors retract this paper. So see that they will come. So they will never come. So then Papier write to those PLOS ONE, and there are volunteers who are writing to the authors, to the letter to the editors, and the paper is retracted. Where is the money come? So this is the another fantastic thing I will show, that this is that another national laboratories in Tiruvananthapuram, and there I'm finding that 2 JBC paper of that earlier director was retracted, and that was retracted with a co-authored with this Professor Rakesh Kumar, who is a major name in the retraction watch. Actually, he lost his job from University of Pittsburgh, and because there was something, everything in those, if you go and the website and you will see, and there were, he sued the university and other things like that. Now, in the DBT Institute website, he is a principal mentor or principal scientist or emeritus scientist who is paying him. And after coming here, when jointly 2 JBC paper retracted, already 2 JBC paper retracted. So if you visit the institutional website, then you will see this, because RGCB institutional website is there. He's a national chair distinguished professor. And so who is paying? Is your and my tax money. So is this list, anyone can find all the retractions from the persons, because this is that US retraction watch against the name of this person, and how his recruitment was done, who is paying him our tax money. So these are, now I am coming because it's the Teachers' Day. I'll not go to the Teachers' Day detail, because there are lots of plagiarism involved with these things also, because those are also available. But here I will show you it is a disaster if you are a teacher. Because as a teacher, I have the dual responsibility, not only to do the research, to show that what is the right and wrong to my students. I cannot fail that. So it's a disaster. If you find here, you see, this is the again that Cleveland Clinic, this first authors are teacher. First author is a teacher now. And you see that this is this thing, that this paper is retracted. You see this is that how these cut pastings and this is same thing doing different. There are lots of clumsy splicing of lots throughout this paper, but I agree this post raised the matters authors could address, while post 3 is pushing matters too far. This is the comment of the Papier. So however, the journal did not agree with the withdrawing author's evidence. The withdrawing author strongly disagree on any duplication between the first 2 lanes of this 3B figure. But ultimately, the withdrawing author stated that none of the results presented in this article were compromised and the conclusions of the experiment remain unaltered. But that paper is withdrawn. So see this, another paper retracted from the same teacher, from the same laboratory, from USA. Another paper retracted from the same teacher, from the same laboratory. You'll see all of those if you go to those websites, and you'll see these are those duplication of image duplication one by one, and then withdrawal of those papers. Same in this site, if you go in those journal site, they are withdrawn. So there is nothing happened, but patient dead. So it's a total healthy situation, nothing happens, patient dead. So those papers are retracted. Now from the individual laboratories, when this teacher is the what I will say, the corresponding author, it started again. And this is as an independent researcher, it should be noted that the authors make use of different size markers to demonstrate the 270 base pair band on the internal control, which is run separately from the collagen RT-PCRs. This control amplicon could practically overlap with the collagen RT-PCR product. There are several errors in the annotation of the subsequent gel, and these are those things when those digitally manipulation is shown beautifully by the volunteers. By adjusting the levels, it is clearly evident that the bands in at least 4 of the panels are not continuous with the rest of the background and the markers. Whatever you say that it didn't do, but it's showing that this is, if you see this pixel size and everything, they are not matching, they are cut pasting. So these papers are also just in the line, they will be retracted. You see that another things from the same laboratories, you find this is this, all of those places there are images concerned about the figure 6C. Several areas within the photos appear to be visible multiple times, as highlighted below with the colored boxes. Could the authors please explain what happened here? So this is that again you are finding. So when you are seeing this is again that same independent researchers, you find in the figure 5C, if you find this, these two are same, these two are same, cut pasted here. 2 years back authors responded, 2 years back, but still nothing in journal website that they say that authors, we have gone through all the concern raised here, we have a raw data related to this figure, we'll be communicating it to the journal editor at the earliest. But I checked in the journal website, nothing mentions. And already this particular teacher has 10 articles in the PubPeer Science. Are you in a position to teach ethics and integrity to the students? No. See, this is that another teacher doing, what is that, another teacher's laboratory. If you find, that is the thing, if you find this is the BSL2, if you find this is the same image, this image same. So this, see this image same, but the figures are, this is C and this is E. So this is clip Caspase 3, this one and this one is same. So this one and this one same. 2 years gone, no response from our side. Serious concern with another data presented in figure 4D. Again one data, that is that flow cytometric data, showing the same areas marked with the same color. Zoom the image to verify, although it is very clear this is the image duplications. So how we can teach? So this is then another thing I will show from the another level, that another teacher from the laboratory. This is the histological slides. Here you will find that volunteer showed this one and this one is image duplications. And instead of going through that, admitting these things, that this person was asking that digitally that you are coming with this thing, which that they are saying that which particular, this work was done when I was old, I was in a postdoc and everything. So like now you are, that the person is saying which software as a volunteer you using to pick up these things. So then that particular volunteer Spathicarpa lanceolata spotted the accidental duplication, say that I doubt that any advanced software or AI was necessary to pick these image duplications. So what are these things? Other scientific integrity related stories, they are full with the Indian Science Administration. These are the observations. I am not just, I will tell that may be coincidental since speculative, these are. But many scientific organizations are CSR, DST, DBT, ICMR, etc. And they are the major grant giving bodies. And the headquarters scientist advisor generally involved in proper project selection procedures and section-wise proper distribution of funds. And they are the yearly review of the grants of constituting proper. So now these headquarters scientists, many places taking advantage of, they are controlling their funds. There you will find some of them appearing as authors in some of those scientific publications of those awardees. Isn't it conflict of interest? And drastic increasing of creativity of publications. When a person is a scientist and now become national laboratory director, within 5 years you will say logarithmic jump in the publications, because now you'll find that entire laboratory of that national laboratory, all of those PIs are putting these national laboratory directors, current directors name in the project. And also having conflict of interest, still sitting in award giving committees, because if you're a student or if you're known, you cannot go, but it's every time it is like there. Sometimes I find that in an award giving committee, I was in a foreign there are foreign scholarship of Indian government. I'm finding the student is coming and my another expert is telling is my PhD student. How can it be? You cannot. And then I said that you cannot be in the room, the person is mad at me. So what are the consequences? Consequences is like that. I'm coming to this NCBS paper, to my younger researchers. You see that distracted article, discovery of these iron sensing bacterial riboswitches. This is the Nature Chemical Biology NCBS famous paper. It started with this. There is a cut mark. So these are surrounded by the square, making it look like this one. You can see it is a perfectly a square kind of thing. It's cut pasted. So I'm not sure how this happened. I believe your concern will find it. Now I'm showing step by step. RT Ramesh is writing. Indeed, there are many figure irregularities. Perhaps someone could volunteer to contact the journal directly to notify. So when this thing is going on, then what she did, Sridhi, dear sirs, thank you for sharing your concern. In response, we have uploaded the raw images as obtained from the instrument so that you can rest your concern and you can see. Now you see all of those images, files, everything, because later she wrote there in the journal site that first author actually that one of the author took all of the data. She doesn't have any data. But there are the paper track in the, if you go to the papier, that she uploaded all of those data and everything. And then the volunteers got the access of those all raw data, and instead of now only making those image duplication, they showed that entire paper lots of those manipulations in the images. And later she was telling, no, first author took all of those data, how can it both can be true? And then those particular volunteers using pixel by pixel all of those plots and data from those given data, and show one by one there are multiple image duplication and image problem. You see, this is the original image, and you see this how it is. Then they come all of those particular this sensor KCL that particular these images, and show there are a problem. So use this. More or less, the readers are saying the GL and blot images in the paper have extraordinary number of irregularities. Authors is saying we have posted raw images. Readers is saying raw images show the exact same irregularities in the published images. Authors is saying images have different timestamps. And volunteer is saying stamps do not explain the irregularities. Author silence. Bottom line, the myriad irregularities uncovered here raise serious concern about the data integrity in this paper. And the author response largely ignored the key issues. And ultimately, this paper was retracted. And now she is saying, we no longer have access to the raw data of these experiments and hence are unable to verify salt-dependent binding affinities of sensi RNAs. Then, in the first time, she submitted all of those raw data. So, from where those raw data came? She deposited the raw data. Now she is telling no access to the raw data. Entire blame is now on the first author. So either she deposited false data earlier or she is like now. So now you understand entire of this pressure will come on the bench worker in the student. And fortunately, immediately papier volunteer checked, and then they showed another image duplication in a JMB paper. And you know that when we do those plate, then actually bottom of the plate we always write, because in the lab if you work you know, because top cover anytime can move in any side. So the agarose gel that containing that particular plate, their bottom you'll write. So now she is saying no, it's probably assuming the labeling in the bottom and other things, but it is not like that. It shows clearly that one thing to realize the mistake in the relabel, it is quite another thing to take photofrost and relabel. Clearly this situation calls for an explanation from the authors, from the same. So now probably that Dolly Mehta will be in the altar. So now what are the consequences? CSR took the first step. They say the statement on the scientific misconduct in CSR laboratories are interim action. Would like to assure everyone that the matter is under active consideration and request to all patience and understanding. So now that CSR decided in all of their national laboratories, there will be data integrity officer, and that person will take care of all of those things. These things are happening. Then this statement was dated in 2019, September 2, more than years, but still we do not know the outcome. But currently one of the CSR scientists told me that the memo came and every CSR laboratories have a scientific misconduct officer, the data integrity officer, who will check and who will regularly do this job. And consequences, retraction, humiliation, because no Nobel Prize laureate in big trouble, Photoshop, Western blots, and other serious issues, about 30 papers. I am so tired of data fabrication. Problems, promotions, job loss, future benefit. Why India? Why doing this thing? Because India's system is award-based evaluation system. Nobody bothered about the science. Nobody bothered about the question. Only JBC and other papers, though my scientific impact is high, so then you will calculate those things. During the Bhatnagar Prize, none of those Bhatnagar Prize committee members going there after reading those papers. They go only whether these people are in my club, or they calculate what is the journal name and the scientific these things. But never big prizes are being allotted in this situation in Western countries. They see what is the science they are doing. If that science being taken away, whether the current position of that area science attain that height or not. And here I'm taking responsibility. None of the committee members read those papers and go to the committee room and do the judgment. They never do that, because our country that one person, when they are becoming big, they are sitting in 200 committees. So first things we'll have to do, that you will have to minimize. One person can sit in one committee, at the most two committees. Then I can blame that person. You go and read during project evaluation or the paper evaluation. So these are the consequences. Consequences, your name will be in the retraction.com. Many more papers are being retracted after mentioned in the PubPeer. And now scientific administration can explanation for misuse of the funding money. Young researchers are suffering more. Any prizes in India giving, now they are checking in the PubPeer. So, but my point is like that young researchers, if they are in fault, they should be punished. But they cannot be punished when committee members are having papers in the PubPeer list. Those committee members' names should be in the open, so people can do their name in the PubPeer and then say that if you do something wrong and something offensive, and then you cannot give judgment on the same offense. Most of the award giving committees are now checking PubPeer. One or two extreme cases, people lost their job. One in IMTech, another, I forget somewhere, I saw that person lost the job. Getting good Western postdoctoral laboratories become tougher from India. Very recently, one national laboratory director didn't get the second stint, possibly because he has 15 papers mentioned in the PubPeer. Prospective PhD students, the bright students who matter most, they are now checking the PubPeer and seeing those persons. Those bright students means they have many other options. They will not join to those IISc and others. They will go abroad. So they will never tag themselves. They will avoid with the labs with the controversial papers. And remedy, now I am talking that what you can do, the young researcher. Forget your mentors, that how far ethically you can modify jail picture digitally. That is my job to tell you, that you can take the picture and you see the best intensity, every resolution you check and do and fridge after proper labeling. Save and lock the image. So never touch that image again. You cannot do that every time sending to those jail, you modify that different. One time you do with every intention, best intensity resolution, proper labeling, and then fridge. No more modification. Or you want to do every time modification, then in the methodology of the paper, write every time you modify. Then if reviewers are agree, you are agree. But reviewer will never agree. So only one time you change whatever you can do with this figure, and they paste it with your lab notebook with corner cutting or with signature, so that never it will be manipulated. You will never be in those things, because these days negative result is also publishable. Negative result you cannot publish in Nature or Science, but there are journals who will take negative result. They have their own meaning. So it is not like that you are not going to getting paper. So never fall into the trap. Never be pressurized if your mentor is saying that I want only this kind of result. So all researchers, especially prospective PhD students, they should and must go through some courses in this direction. And now I feel some of the faculty should attend this kind of seminar and hand-on training on image modification, not manipulation. Mentors should and must develop maximum time with the students, because your job is to mentoring with them. Your job is to show what is right and wrong, and then students will swim and decide. Must sit with the raw data. If you are very busy also, then at least once a week, never blame to the bench workers. If something come, then mentor should take the onus and responsibility and say only I am responsible. I'll show one case. It is not like there, because NCB is also saying no, every blame is on first author. Award-based evaluation and promotion must be stopped immediately, or we need to change the perspective. And committee members, they should and must read the papers of the projects before attending the decision meeting. Nowadays, that is not the norm, and majority being done in the club basis. You belong to my club, or you belong to, we have one Cold Spring Harbor meeting, Guho Research Conference. All the top people are going. It decides there that who is with whom, everything club is there. These are demanding jobs, especially award and project award committee members. So one individual should not sit in one, or at the most not more than two committees. Otherwise, they cannot do justice of their job. Blatant manipulators should be denied the research grants at least for 5 years. Institution and university should deny research students to them, because if they are doing that, they are not in ethical position to teach students, at least for a moratorium period. As a mentor, another open thing is like that. These days there is a bioarchive, is a boon. So anything as a mentor you can publish there. As preprint papers are hosted into the website, they are not peer reviewed, but undergo basic screening and checked against the plagiarism. So if after 1 week or 2 you get those things, then you check those things and send to the right journals. Or many a times you get good review, so then change the paper according you say. So bioarchive presentation immediately put, and this is already that if you have a you are afraid of to be scooped, it is the very good area bioarchive you can publish. If you find something is not appropriate in an already published or accepted papers, as a mentor or a corresponding author, take the full responsibility and gracefully retract the paper at the first chance. Please do not find against evidence. I will show that how it will happen. Please do not complicate the case further. Next case I will show. As a mentor, please do not publicly humiliate the bench worker like R.T. Raman did with the first author, since you are also laxed your fundamental duty to supervise that person, him or her, properly. That is also unethical. That also I will show you now, right now. So when ego hurts, this paper is clearly image duplication. Although I then at the time say that I hope the authors, the volunteer said, hope the authors could clarify showing the original photographs petri dish and with the whole petri dish not that is cropped. And then this person says that principal investigator, no, this is doesn't make sense. First I have to do plenty of serial dilution spot test, and I'm not aware of anyone who does the replica plating of them, as noted by original pictures of the whole dishes school. And this person is very powerful, so there are lots of manpower, so they did all kind of forensic and everything. There are at least more than what I will say, that 20-30 threads, comments and other comments. This person with the ego hurts, nothing saying. Then I see sometimes after fighting so much that 30-40 times with all of those lackeys around, and then there I see in the website of that microbiology, there has been an erroneous unintentional duplication related to the two top rows of the left and middle panels. Given the age of the article and the SCM lab has been closed, we no longer have the original images. We note that potential duplication does not impact the conclusion of the paper. Then why you fought? Because your ego hurts. And now I'm showing that how that onus is always on the first author. This paper from the Indian Institute of Science, the authors, is a two-author paper. The authors of this paper would like to retract their article. Retract the article, fine, retract it. Then saying the first author takes sole responsibility for this misrepresentation. First author is a young postdoc or a young PhD student, his or her life on the line. This is a two-paper author publication, most famous Indian Institute of Science, and already retracted. Instead of only taking the responsibility of the retraction, mentor is pointing out only the bench worker. During the entire process of the publication, which I showed earlier, what the mentor was doing, sleeping. As a mentor, you cannot desert your students, postdocs, bench workers. As a mentor, you can repeat a couple of major experiments by another student. So long time ago, the famous Chinese philosopher told life is really simple, but we insist on making it complicated. So why? That why first author? So because this is that one heading that I saw in the Times magazine with a bribery rate of 39%, India worst in Asia survey. So this is not an isolated case only in the science and technologies. Everywhere we are going through that. So I will finish now. Science is not a thank you. Silence is not an option. That somebody will have to say what I am doing. In the process, I have many enemies. And from the process, I'm happy. So 200 places must be my all entire CV being downloaded. All my papers are ransacked. So I can say, you didn't get anything, you will not get anything. Thank you. If you have any questions, I will answer it.
Thank you, sir, for sharing your views with us. And we have two questions. Sir, would you mind taking them now?
Sure.
Okay. So the first question is from Vijay Vishwas. I think you have already answered it, but again, in your opinion, what would be the correct strategy to mitigate this problem, and what about other kinds of data fabrications?
Okay. Dr. Bishwas, I already told about those strategy, because strategy means that I'm not in the administrative part or I'm not involved in administration anyway. I already pointed out the strategy. First thing is like that in India, award-based evaluation system should be immediately stopped. And I'm actually against that your job will be gone and other things like that, but there should be some checks and balances. That means that you will have to go through some ethical classes, And if your papers are in there, so if it is a university or in a national laboratories, that you will not get student to mentor for at least for a moratorium period, that you will debate and decide whether that will be 1 year or 5 years. And if you are using government money in our country, more than 90% fundings are from government agencies, BST, DBT, Department of Energy, CSIR, wherever is possible. So this is our tax money, hard-earned money. So when we are getting from the money from the government cover, the funding should be immediately stopped at least for 5 years. So these are the things, and particularly the people who are marred with this. I didn't say that I'm making another slide. The national laboratory directors, most of the national laboratory directors in the biological laboratories are having this paper. Right now, this tide is changing. So if you have that kind of paper, you cannot be HOD of a department. You cannot be the director of a national laboratory. Those are the checks and balances should be done by our science administrator. But the problem is like that our science administrator most of the time are having manipulated paper in the papier side. So that's why nothing is happening. So it's the young researchers only they can say, I will not go to this laboratory whose paper in the papier. And that is the first checkpoint, and voluntary you can do, all the young researchers, and you can spread this throughout India, that wherever you will find a laboratory is doing this thing, no matter what, I will not join to the laboratory. That we can do. And other kind of data fabrications, obviously these are the digital era, whatever those digitally possible they are doing. But right now I am finding there is a wave of other kind of fabrication things are coming in this thing. But interestingly, if the data fabrications happening in physics, chemistry, or specifically in the mathematics, is not data fabrication. Maybe the problem with the data interpretation, because we are unable to properly, but that is not a crime, but there are lots of fighting in those of physics and mathematical paper in the thread. Some paper in the papier, there are 129 comments. But other kind of data fabrication, slowly this apparatus is also improving. So fabrication will be there, that and they will be catch either by today or by tomorrow. Right now, these image duplications and others, we are capable of doing that, and very soon others also we will start doing that. Those kind of experts, physics expert, chemistry experts, and others, they will be there. Don't worry. Next question, please.
Thank you, sir, for your explanation. The next question is from Monish Mandhotra. And sir, how a student get knowledge about such unethical peer review authors in India and abroad? As you mentioned about PubPeer, what about others?
There are two websites, pubpeer.com and Retraction Watch. You can go and see that they have a patience, and see that whatever the laboratory you want to go, you search with their name. But in the search, you will, if it is a very common name, then you will have to go. You'll have to spend some. If it is named like those Amit, or if you name like those Samit, or this kind of common name, or Malhotra or Agarwal, there are lots of those things. Then you will have to go one by one, and from their queue you will have to go to those paper, and in the paper you will get also address of those authors and all of those things. But you can figure it out that where you want to go, what is the position in the PubPeer. And I say it's a random process. Every day 30, 40, 50 papers are coming. So it's not like that a lab is looking very clean now. Maybe in the down the line that paper will be there. But whatever is already there, it's easily you can find out going to the PubPeer or the Retraction Watch.
Thank you, sir, for your explanation. So that was all the questions that we have today. And thank you again, sir, for speaking in our platform. We are really honored.
Okay, thank you. So it's a that it's always that people should know that what is going on. Just they will have their own judgment now, that what is going on and how they will cope up with that. Specifically, young researchers, they are in the line. So always, as a mentor, I can blame that everything done by my first author. So, but that is not the escape. As a mentor, I cannot take the route. But that judgment is yours. So you will have to decide, and you will have to go where you will have to go. Okay, it's your life.
Yes,
Okay,
sir.
thank you.
Thank you,
So
sir.
no
Thank you,
other
sir.
questions. Okay.
No, sir. No
Okay.
questions.
Okay.
Okay. Thank you, sir.
Thank you, Akashlina, for hosting me. Okay. Bye.
Bye, sir. Bye. And to all our audience, uh, thank you for staying with us. And do check out our website and our YouTube channel where uh, all our previous talks are archived in. And follow us in Facebook for the latest updates about the upcoming interesting lectures. And do join us for our next session with Professor Rajgopal Dhar Chakraborty. And till then, stay safe and have a nice time. Bye.